Hi, everyone. I hope you all can hear me, but welcome to today's coastal state discussion. When we're looking at changes in the Benthos. My name is Meredith Haas, and I'm from Rhode Island Sea Grant, and I will be your moderator for today. And for more information on the coastal state series, you can visit our website. Let me get out of the way. You can visit our website where you'll also find recordings from past discussions if you're interested in that. But visit us to get updates on upcoming events, or also feel free to subscribe to our newsletter and you can get those updates directly sent to you. And today we're going to be focusing on changes in the benthos and kind of looking at how these benthic ecosystems on the seafloor of Narragansett Bay have changed over time, looking specifically at the impacts of warming water temperatures and changes associated with nitrogen reduction efforts from wastewater treatment facilities, and what those changes mean for the overall health of the bay, as many of these organisms are sedentary and so are some of the most vulnerable to local changes and therefore identified as key indicators of overall water quality. And so for today's, let's see, I want to thank the sponsors for today's discussion, the University of Rhode Island's Graduate School of Oceanography and the Nature Conservancy. And before I introduce our speakers and let them get started, so you can hear about the things that, for the reasons why you're here, I just want to give an overview of today's format. We're going to split up into two different sections. We're going to have 1 hour presentation in total for the speakers to present, followed by an hour Q&A session. So you can share your questions either using the Q&A option or in the comment area below. And so we'll circle back to those during the Q&A. And you'll also be able to raise your hand if you just wish to ask your question directly. We'll also be recording today's presentations and discussion, and that will be uploaded to Rhode Island Sea Grant's website. I'll do that as quickly as possible, but you can expect that either later this week or next week at the latest. And so with that, I'm gonna scooch on out. I want to thank and introduce our speakers for joining us today. Dr. Jeremey Collie, who is a marine ecologist and professor at the Graduate School of Oceanography, who's joined by his former graduate student, Shaina Harkins, who focused her thesis on understanding the drivers of these changes in benthic communities in the Bay, which I think will help, has helped start establishing a new baseline of ecosystem functioning that will have implications for management practices. And we'll also hear from Heather Kinney, right? Am I saying that right? I think so, Kinney, who's a coastal restoration scientist from the Nature Conservancy. And her work complements Dr. Collie's and Harkins, but looking at benthic communities, specifically in the Upper Bay and Providence River regions, where nutrient loading and low oxygen conditions are historically more prevalent. So yeah, I just want to share, just want to thank you guys for joining us. And I think, I set that up. If Shaina, I think this is now being handed off to you. If you kind of either want to clarify any of my summary, but get us started in kind of what you guys are working on.
I think you gave a great, concise, all around summary, Heather, or sorry, that's Heather. Yeah, Meredith. And so I will start us off by sharing my screen and yeah. So, hi everyone. Thank you very much for coming to the Coastal State Lecture Series. My name is Shaina Harkins, and this first talk is on the response of benthic communities in Narragansett Bay to changing nitrogen input and temperature. So the first question then is, how has nitrogen and temperature changed in Narragansett Bay? So beginning in the 1970s, federal and state regulations mandated a decrease in nitrogen input into the bay from wastewater treatment facilities. This was due to eutrophication, or the increase in the rate of nutrient input into the bay, causing overproduction and over-respiration combined with weak summer stratification and the occasional weak spring neap tide, preventing tidal mixing that replenish bottom water oxygen, leading to areas of hypoxia and occasionally anoxia. On top of this, in the last 50 years, surface temperature has increased 1.6 degrees Celsius. Benthic organisms can be used as indicators, as Meredith said, to monitor the overall health of the bay, as hypoxia actually begins below the sediment, because that's where, tidal mixing starts to affect it first. You decrease the level of oxygenated sediment to the point where there is no oxygenated sediment, and then hypoxia starts to take over the bottom layer of the bay. And we actually happen to have a wealth of historical data of benthic abundance in the bay. So this is one of my favorite images to sort of show how regular succession occurs for benthic organisms as a response to a disturbance. So benthic organisms, or organisms that live in and on the sediment, have a successional response to disturbance. Starting at the disturbance, and actually I think I can get a laser pointer in here, sweet. So starting at the source of disturbance, you have a layer of oxygenated sediment that is either very thin or non-existent. So what you have are high abundance, small, short-lived, opportunistic species that are found at the sediment surface near the pollution source, such as tube-dwelling amphipods and a couple of capitellid and scolelepis polychaetes. The further from the disturbance in both distance and time, the more the oxygenated sediment layer increases. So lower abundance, larger, deeper dwelling, and longer lived organisms begin to replace the opportunistic species as the oxygenated sediment layer increases. So given that background, the hypothesis of this project was the mandated decrease in nitrogen input to Narragansett Bay will significantly change the macrofaunal community composition of the upper bay through time, with temperature as a secondary driver. The project had three main objectives. We wanted to assess the current macrofaunal community of the Western Passage at a northern upper bay station, a mid-bay station, and a southern lower bay station. We secondly wanted to assess the current epifaunal benthic community at the same three stations. And lastly, the most important, we wanted to assess the effect of change in nitrogen, temperature, and other environmental variables on the change in the bay's macrofaunal community at 3 points in time: the late 1970s to the mid-1980s, the early 2000s, and the current sampling from 2021 to 2022. Sampling occurred in June of 2021 and 2023, as I said, from the URI's Captain Burt. We had three stations throughout the bay, one near Conimicut Point, which was our upper bay station, one north of Jamestown, which is our mid-bay station, and one outside of the bay proper in the lower bay in Rhode Island Sound. For each station, we took 15 replicate diver-collected cores to target infaunal macrofauna, or the organisms that live in the sediment, and 2 replicate beam trawl tows, which targeted epifauna, or organisms living on the sediment. Large organisms were identified, measured, and released on the boat, while small organisms were taken back to the lab to be identified to the lowest possible taxonomic group and measured on a gram or microgram scale. For our first objective regarding current macrofauna in the bay, we found an overall decrease in both abundance and biomass from 2021 to 2022, with the most notable decrease in Providence River. So you see, in Providence River is in red. Next, we have what are called ABC plots, or ABC curves. They're abundance biomass comparison curves with cumulative percent dominance abundance in green and cumulative percent dominance biomass in blue. As I mentioned, higher successional stage benthic communities contain large biomass organisms with low abundance, dwelling deeper into the oxygenated sediment and living longer. A high successional stage benthic community would be expected to always have a biomass curve above our green abundance curve. And the W statistic, it's a little small, but it's right here, is a measure of how high the biomass curve is above the abundance curve. And you can see from these plots that all three communities have biomass above abundance with a W statistic greater than 0.6. This suggests that the communities at the three stations in both years are at a higher successional stage. This plot is known as a multidimensional scaling plot, or MDS. It plots the replicate communities for each station against each other in a non-scaled plane to show how they lie with each other in similarity. The closer they cluster together, the more similar the replicate communities are in composition and abundance of organisms. North of Jamestown is in green, and Rhode Island Sound is in blue. Both cluster together well for both years. While the closed red triangles for Providence River in 2021 cluster completely apart from the open red triangles for 2022. This suggests that Providence River community is distinctly different year to year. So ANOSIM, or analysis of similarities, compares the community composition between replicates of each station to community composition between stations to see if the difference in community composition is significant between stations. Both pairwise differences, so Providence River versus north of Jamestown, Providence River versus Rhode Island Sound, and north of Jamestown versus Rhode Island Sound, were significantly different from each other, as was our global difference. And then we have our best analysis. Best analysis compares the change in environmental variables to the change in community composition to see if there is a correlation between the environmental change and the change between stations. The environmental variables found to be most correlated to community composition changes between stations were dissolved oxygen, temperature, sediment percent carbon, and sediment percent nitrogen. And actually, if you look at how these environmental variables change throughout the bay, it's pretty obvious that what we're following here in this best analysis is the environmental gradient that we see throughout the bay. Bottom temperature decreases, dissolved oxygen increases, and sediment percent carbon and sediment percent nitrogen both decrease as you go further away from the developed Providence area to the less developed lower South County area. So what we can conclude for the first objective then is that the three communities sampled throughout the West Passage represent three distinct high successional macrofaunal assemblages. We also noted that year to year, Providence River distinctly changed in community composition, while north of Jamestown and Rhode Island Sound were more similar year to year. So I'm just going to take a short palate cleansing break and show you a couple of worms that I found. This one on the right is called, colloquially known as the broomworm because of the bristles on its head. Second objective was current epifauna at the three stations. Keep in mind only 2 replicate beam trials were performed. So notably, north of Jamestown abundance increased quite a bit between sampling years from 2021 to 2022, while oddly, biomass decreased year to year for all 3 stations. This actually suggests that some of the assemblage for north of Jamestown for 2022 is probably made up of an abundant, small, opportunistic organism, this guy. So the culprit for our high abundance in 2022 was actually a worm named Polydora cornuta. In 2022, one of our beam trawls dragged through a Crepidula fornicata or slipper shell reef, and within the shelf of the slipper shell right here, full of sediment, it's the perfect space for a small opportunistic sphenoid like this to colonize. Much of the abundance for north of Jamestown of 2022 was actually this worm specifically. Our ABC curves for the epifaunal communities suggest that the 3 stations in both years were high succession, as the biomass curve for all 6 plots is above the abundance curve, and our W statistic for all 6 plots is greater than 0.6. Now, our multidimensional scaling plot for epifauna is a bit less telling. As I said, we only have 2 replicate beam trawls, but what we can gain from this plot is that Rhode Island Sound clusters far apart from both north of Jamestown and Providence River, which cluster much closer together in similarity, and they actually cluster apart from each other in similarity from 2021 to 2022. Actually, in 2021, the winch wire for our beam trawl was not long enough to drag along the bottom for Rhode Island Sound, so this accounts for our large difference in similarity. Again, ANOSIM is less telling for epifauna due to replicate. As you can see, our p-value is quite high for our pairwise comparisons, but our global statistic is significant enough to tell us that the Providence River north of Jamestown to Rhode Island Sound stations for epifaunal communities are significantly different. For our best analysis, the 2 most correlated variables were temperature and salinity. And again, this more points towards our environmental gradient in the bay. And I think what's more telling about this best analysis result is that sediment and nitrogen percent carbon, sediment percent carbon and sediment percent nitrogen have nothing to do with epifauna because they live on the sediment. So they're much less impacted by the contents of sediment because they live on the sediment. So what we can conclude from our second objective was that the current epifaunal communities at the 3 stations are distinct, high successional assemblages. We saw a decrease in biomass at all 3 stations between years, with a large increase in abundance between years at north of Jamestown due to the sampling of an ephemeral reef containing a colony of small opportunistic worms. And for our next palate cleanser, we've got some decapods that have some egg clutches. And these were both caught in the bean trawl. Our third objective and the last objective was to look at the effect of changes in nitrogen, temperature, and related environmental variables such as dissolved oxygen, chlorophyll, and epifaunal abundance as the change in macrofaunal community through time. As I mentioned in the beginning, a wealth of historical data exists for benthic community abundance. Some studies use the same methodology, so our diver collected cores, which are the studies we are going to be targeting we did target for our quantitative comparison to the current results, while others did take different methodologies, which won't be considered quantitatively for comparison. First, we're going to look at specifically the most directly comparable and most recent historical study done by my collaborator, Chris Calabretta, and my co-PI for the project, Candace Oviatt. This was done from 2000 to 2010, beginning right before and during the decrease in tertiary nitrogen removal was mandated. Chris aided in ensuring the methods were replicated as closely as possible and in taxonomic identification for my study. The data for the early 2000 study was compared to the current study 7 to 8 years after the decrease in tertiary nitrogen input was completed. The previous study only had data from our upper 2 stations and not Rhode Island Sound, so that will be ignored for now. This multidimensional scaling plot shows Providence River communities as red dots and north of Jamestown communities as green dots. North of Jamestown, you can see clusters fairly well together in similarity, and so does Providence River for the early 2000s. When you consider the 2020s, Providence River becomes much more similar to the north of Jamestown dots, and actually most similar to the 2020 and 2021 and 2022 north of Jamestown communities. Our analysis of similarities shows significant dissimilarity between the studies themselves and the stations. Our best analysis showed the highest correlation with nitrate and nitrite, which were components of our nitrogen measure, dissolved oxygen, and our second best analysis revealed a secondary correlation with temperature. So if you look at the time series for environmental variables, it sheds a little more light on what's going on environmentally. The early 2000s saw a steady increase in dissolved oxygen, ending much higher in our 2020. And then the varying concentration of nitrogen, that's a little less explainable from this plot, but it's still correlated. And then our average temperature showed an overall increase for actually both stations. So from the comparison to the early 2000s data set before the tertiary nitrogen removal to the 2021 to 2022 data set 7 to 8 years after tertiary nitrogen removal, we can see that nitrogen correlated the most with change in macrofaunal community at the 2 stations, and dissolved oxygen and temperature were secondary correlations. Okay. We also found that the Upper Bay Providence River community has become more similar to the Mid-Bay north of Jamestown assemblage over time, a historically high successional stage community, the north of Jamestown community. This suggests that the Upper Bay station has increased in successional stage over time. And for this palate cleanser, we have a resident sea cucumber. They are not originally pink. Samples were dyed pink with rose bengal to aid in sorting and identification. Lastly, for our third objective, we expanded our historical environmental variable comparison to include the late 1970s and early 1980s, those studies that employed diver collected cores. The peak of nitrogen input occurred during the sampling of the historical diver collected cores from 1975 to 1986, compared to right before tertiary nitrogen removal began in the early 2000s. And then currently 2021 to 2022 sampled 7 to 8 years after tertiary nitrogen removal was completed. Again, only the 2 Upper Bay stations are being considered for our historical environmental comparison. Rhode Island Sound is being omitted. It should also be noted that north of Jamestown was sampled much more frequently than the Providence River Station. I would like to reiterate the benthic successional response to a disturbance, and that this response occurs through space and time. So again, you see higher abundance, opportunistic, short-lived species that burrow at the surface sediment in low oxygenation, and then as the oxygenated layer increases, you see longer-lived, low abundance, higher biomass species that burrow deeper. So this is a pretty involved plot. So this is why I drew some arrows to follow the timeline, and just keep in mind that open circles represent Providence River while closed circles represent north of Jamestown. So beginning in the 1970s, there was quite a bit of similarity between north of Jamestown and Providence River, and then it veers off. Providence River has a bit of a gap in our sampling that we are comparing to, but in the 2000s, it switched to the other side of the plane that we are plotting on, and then it settled in the middle over here, nearer to north of Jamestown in the 2020s. The north of Jamestown community, again, starts nearer to the Providence River community in the 1970s, but then shifts to the middle, where it sort of stays pretty stable through the 1980s and 2000s. And again, it settles out nearer to the Providence River Station of 2021 and 2022. So again, you see an increase in similarity to, it does its movement, but it ends up being fairly similar to north of Jamestown when it settles out in 2021 and 2022 sampling. Our analysis of similarity revealed significant dissimilarity between stations and even more dissimilarity between years. That's revealed a less significant but still present correlation with ammonium, another nitrogen component that was different from our nitrogen component of our last historical comparison. That was nitrate and nitrite. This is revealing ammonium. And then if we went a little further with our second best analysis, the second best correlation showed a correlation with temperature as well. So then if we look at the environmental time series that go along with our data, we can see that total dissolved inorganic nitrogen and ammonium both decrease through time. Temperature increases through time. And then, again, the nitrate and nitrite component also mirrors overall DIN decrease. So then we can conclude from our last objective that nitrogen was the most correlated environmental variable contributing to the change in Upper Bay macrofaunal community, with temperature as a secondary driver. And this just sort of reiterates what we found in our first historical comparison to the early 2000s, but it expands our timeline and gives us more data to work with. We did not have dissolved oxygen data that went back this far, but there would also probably be a correlation to dissolved oxygen if we had the data that far back. The other conclusion this comparison reaffirms is that the Upper Bay Providence River community has become more similar to the north of Jamestown Mid-Bay community over time. The north of Jamestown community is historically considered a high successional community, suggesting the Providence River community has increased in succession through time. This is a mantis shrimp we caught in the bean trawl. So then finally, our conclusion overall can be, despite uncontrollable variation due to climate change, the results of the current study represent successfully managed nutrient reduction, improving macrofaunal succession, abundance, and biomass in the Upper Bay of a historically polluted community. And with that, I would like to thank everyone who's involved in this project, all of my co-PIs, Jeremy, Candace, Austin, Connor, and then my collaborators, Chris and Steve, all of my undergrads and all of my lab mates, all of the divers that helped, and the captain of the ship, Steve. So with that, that's it. So yeah.
Great. Great. Thank you so much for that. Again, for those of you that have any questions or comments, please leave them in the Q&A or under the chat, and we'll circle back to those. But now I'll hand this off to Heather.
Great, thank you. I'll share my screen next. All right. So hi, I'm Heather Kinney. I'm the coastal restoration scientist for the Nature Conservancy. I just wanted to say first that the analysis for this work is still underway. So I'll be sharing about methodology and some examples from what we've collected. But first, just want to give a quick thank you to folks who've been helping out with this project, even through a pandemic and equipment failure, and folks who lent us gear and data, particularly those from the Narragansett Bay Estuary Program, Narragansett Bay Commission, EPA, and Department of Environmental Management, as well as others. So thank you. And yeah, today, so we'll be sharing about our project, Benthic Communities and Habitats as Indicators of Changing Water Quality and Ecosystem Health in Upper Narragansett Bay and the Providence River Estuary. So I made an assumption with the group joining today that folks know where Narragansett Bay is, so apologies if you aren't from the area, but our project was focused in the northernmost portion of the bay, specifically the areas labeled as the Upper Bay and Providence River Estuary here. And I'll have a better site map in a minute, but just wanted to orient folks. The Providence River Estuary is an urban estuary running through the state's most densely populated areas. And like most urban estuaries, they face their fair share of impacts from human use, which I'll briefly touch on again more in a minute. I'm not a native Rhode Islander, but I've been here for over a decade, and I've spent 6 years at the Nature Conservancy studying this section of the bay, and truthfully, this area has become really very near and dear to my heart. And I say this because improving the health of these systems is so important, not only for human and ecosystem health, but also for all of the culturally significant value these types of waterways hold. So today we're here focused on discussing the biological changes of the bay, concerns about nutrient excess or reduction, and the challenges of quantifying the impacts of climate change at an ecosystem scale, which is huge. But we're here doing that because this area is so important to so many people, and likely many of us who are on the webinar. So thanks for joining. And I wasn't sure how much background we were going to need, and Shaina, thank you. I think the background you provided was really helpful. So I'm just going to do a very quick overview that stretches through kind of a wide chunk of time. So afterwards, we can, correct me if I skip anything or make any incorrect statements. But the area is the ancestral land of the Narragansett, Wampanoag, Nipmuc, and Pokanoket tribes. And before European colonization in the 1630s, I did not check myself, but I think that's true in Rhode Island, the Narragansett tribe and others were said to have a well-established population and were utilizing the river for transportation, trade, stewarding the fish and shellfish and other estuarine resources of the estuary. And the same use of this waterway continued post-colonization. And as European settlement grew, this growth led to increased land development, shoreline hardening, industrialization. More people led to increased pressure on the system, which led to polluted stormwater runoff and contamination by sewage effluents, which, among other things, ultimately changed the biota and health of the waterway for centuries to come. So technologies improved and the population growth stabilized. Wastewater treatment facilities made huge strides in reducing the amount of nutrients going into the water. The degree of reduction in nutrients necessary to improve the system at the time wasn't necessarily known. And again, people didn't necessarily know how this reduction, which ended up being around like 50 to 60%, would impact the system as a whole. But it was needed to help improve the current state of the waterway. And here we are, decades later, still working on measuring and quantifying those impacts, and also including climate change into that, which complicates things, certainly. One reason why estuaries are important for people is because they're a critical nursery environment for juvenile fish. foraging fish species, birds, turtles, just wildlife in general. We're focusing again on the benthic environment today, but many of these species rely on the benthos for food and shelter, even if they do live either just on the surface or in the water column. And we can learn a lot about the health of the estuary through studying these organisms in combination with the smaller epifauna and infaunal creatures, which Shaina highlighted also in her presentation. And here are just some of the juvenile fish species and some tropical ones that we catch up in the Providence River. Also critically important, another reason it's critically important is for commercial and recreational fishing. So the larger adult stages use this water body as well and are impacted by ecosystem shifts and changes in climate, which affect us too. So we had 3 main objectives with some similar overlap. And today I'm just going to share about objective 2. There already were some, like a good deal of baseline information from previous studies. Again, like Shaina mentioned, but we're using a new imagery method, at least new for this area, and a combination of different imagery methods. And in order to assess the benthic community. And another goal that we had was also to compare the use of these different imagery methods to provide insight on balancing trade-offs between data resolution and spatial extent, because oftentimes those two things are at odds with each other, and needs can be dependent on, like what you're collecting that imagery data for, whether you need higher resolution or greater spatial extent, and where to lie in between. So what were our methods? We used 4 different data collection methods. 3 of them were for image collection, and the Ekman grab was the 4th. And that was really for some image ground truthing and to provide a higher level of information to some of the image analysis that we were doing. So we really weren't quantifying biomass here, just trying to get a peek at what was below the surface. So the first is the video sled. So the Nature Conservancy, in partnership with the Department of Environmental Management, collected benthic video transect data from 2016 to 2018. And the Narragansett Bay Commission has been collecting benthic imagery data, I believe, since 2014. And they were kind enough to let us analyze their 2022 video data, the transect data as well. But the sleds are outfitted with a sea viewer camera. They have lasers measuring so that we can measure what's in the frame, lights and water quality monitors as well, measuring dissolved oxygen, salinity, temperature, and depth that the transect was taken. The field of view for analysis for these is about a square meter multiplied by the distance of the tow. So these are towed behind the vessel. And those ranges were from 1/2 to 2 kilometers in length. And for analysis, we analyzed the images at 1-minute intervals. So in order to kind of compare the methods, because the drop camera is a still image, we were just taking images at 1-minute intervals and analyzing those. And on the right are just 2 examples from snapshots taken from the TNC and DEM sled from 2018. So Conimicut Point is a southern site kind of at the base of the Providence River estuary, and Bishop Point is in the Seekonk River, so northern estuary. Next is the drop camera. It has all the same pieces as the sled, except it uses a GoPro for the camera and can take either still or video footage. The area it covers is about a square quarter meter. And for this, you would take multiple drops within a 50 square meter area and get the images you see on the right here. Again, this is the same locations, but Conimicut Point and Bishop Point just using different video methods. And next is the sediment profile imagery camera, or the SPY camera. It has the smallest field of view and the highest resolution of images. And again, the pictures here are colored, so they don't actually look like pink or yellow to blue like it shows in this picture. That's just showing the difference in oxygen layer. And we use EPA's drop camera, and like I said, it has the highest resolution between the methods and provides valuable information for what's beneath the surface that the other camera methods really can't do. And it does this by collecting imagery as a cross section of the substrate. And unfortunately, after 3 seasons of trying it in the field, we were only able to get 2 single photos. And this year we tried and failed for the last time, but the camera itself had a good 19-year run before us. So unfortunately, we don't have this data set to incorporate into our analysis, but sometimes that's how science goes. Lastly, we have the Ekman grab. I already kind of mentioned this, but it was a small Ekman grab, about 1/10 of a square meter sampled and at select sites. And then we sieved down to about 0.5 millimeter. And Dr. Niels Hobbs, an invert biologist at URI, analyzed the samples and ID'd them for us. And we, like I said, collected them at only select sites, which I'll show in a site map in a minute, and at the same time as our drop camera image collection. And that allowed us to get a little bit more of a direct comparison between what we were seeing on the surface and what we were actually, like what was actually underneath. So this was our sampling schedule. The asterisks represented the failed SPY days and a site map with the video transects from Narragansett Bay Commission's 2022 data set, which is being analyzed alongside the drop camera and Ekman data. So we did try to do some multi-gear sampling, which was just concurrent video sled and drop camera collection, but scheduling and equipment struggles also made that pretty difficult. But here you can see we had just the drop camera sites in the dark green. The light green are also just drop camera sites, but they were intended to be coupled with the SPY camera. And then the light blue are the drop camera and Ekman grab sites. So for the analysis, so we've collected a bunch of these images, and we're using the Coastal and Marine Ecological Classification Standard to analyze them, which is a federally endorsed and standardized classification system that provides a universal language for scientists. And this allows, promotes uniformity across research, across management and restoration practices. There are 4 main components to it: a water column component, geoform, substrate, and biotic component. The ones that we've really focused on were the substrate and biotic components, though we did have some water column components as well. But the substrate component is just the character and composition of the surface and near surface substrate. The biotic components make up the assemblage of benthic and suspended or floating organisms. And again, we really picked it because it was standardized and uniform. It provides common language. By using CMEX, we could directly compare it to other research that had been done in this specific area, and also in similar environments or in different environments, but with a similar method. So there's an easy way to compare. It's a hierarchical system that allows reviewers to get as specific as they need to as well, or can be based on image quality. So as specific as you can get if the images aren't, they don't come out that well. And that structure is just represented by the lists here. So we are using CMEX to define biotopes based on the images we collected, which will allow us to compare to previous studies, like I said. And we end up with a dominant space descriptor with co-occurring elements. So on the right is one of the photos taken with a drop camera, and on the left is the CMEX description of the images taken at this site. So I kind of got into this before with the methods, but we took multiple samples or drops at each of the sites in a 50 square meter area. And so this descriptor takes all of those images into account. And with this image quality, we can identify down to the subgroup, which is Crepidula or slipper shell hash or rubble. And the biotic component is Crepidula reef with co-occurring elements, including trace to sparse amounts of redbeard sponge and boring sponge, and Granelia algae, and some evidence of a clam bed. And so I kind of circled these different things here. We've got redbeard sponge, some Cleona growing off of some shell here, a very small little clam siphon, another clam here, and then some Greenellia algae. So this also shows one of our 2 spy images we were able to get. And so you can see the added benefit of having that image for a community succession analysis, since you can see the oxygen layer in the substrate and get a better glimpse of the infauna below the surface, though this can be done with the surface level imagery as well. And you can see some hermit crabs in there too, which is cool. Here's just another example comparing the imagery of a drop camera and video sled, as well as the addition of the Ekman grab sampling. So the image on the left is a drop camera photo. The middle is a snapshot of one of Narragansett Bay Commission sled transects, again, which are being analyzed at 1-minute intervals. And the table and images of what was found in the Ekman grab is on the right. The substrate descriptor for this site is mud to sandy mud with trace amounts of shell hash. And the biotic descriptor is small surface burrowing fauna and small tube-building fauna, trace to sparse amounts of Neceridae, mud snails, slipper shells, and hermit crabs, with filamentous and sheet macroalgae. And then with the Ekman grab, we can get more specific and say that the burrowing and tube-building fauna likely included at least these species captured. As you can imagine, it's really hard to tell different surface burrows apart in an image, so the Ekman grab provided that additional information. And like I said, depending on needs, knowing that this area is characterized by mud and soft sediment fauna at a stage of 1, possibly stage 2 community succession level may be enough, or perhaps a higher level of detail is desired, which a direct sample or spy image could provide. And then this is what I was referring to earlier with the trade-offs for different methods. This is a draft table, but it gives a general overview of the different imaging methodology and how they rate against each other. And so the rank types that we included are the technical expertise to build, which for the spy camera is the highest, and it goes down to the video sled and then the drop camera. And then the technical expertise or training to operate, again, the spy camera being the most complicated, then video sled, and then the drop camera and video sled were about even, but drop camera was slightly easier because it's just a smaller frame. The frame cost doesn't take into consideration the camera used because that can change, and it also was very different based off of the 3 that we were using. But the spy camera, again, is the highest cost by a significant amount, where the benthic video sled runs about $700 to $800 to build. Drop camera is about $150 to $200 for building it, so much cheaper option. And then we included cost to operate, which is just focused on personnel. You do need more folks to run the spy camera, less for the video sled, and about the least amount for the drop camera, again, because it's just a smaller frame and easier to operate. And then the rest of the cost as well were about the same when you kind of take away the rest of the differences. Once you have it built and once you've got the way to get it out there is really just using the boat and the fuel at that point. Though you can use the drop camera and spy camera from shore. The spatial coverage per sample is much higher with the video sled. Like I said, it's 1 square meter multiplied by however long that transect is. You get a much larger area, but we do sacrifice some things, which I'll go into in a second. By doing that, the drop camera was next, and then the spy camera had the lowest spatial coverage. But I'll skip down to the sample detail and image resolution because it has the highest resolution. So the smallest area, highest resolution, and that's kind of the opposite of the benthic video sled. So you can get a larger area, lower resolution. And then the average sample quality also differs between methods. So you get a much higher quality image with a spy camera and kind of lower with the drop camera and video sled. But it may be something that you don't need that high of a resolution, but you want more spatial coverage. So this list just provides kind of a rank to make those decisions. And so for our next steps for objective 2, they're really to complete the remaining image analysis and compare to the same locations that have been previously sampled, including this Narragansett Bay Estuary Program comparison of SPY images from 1988, 2008, and 2018, which used CMEX biotopes. And here is just an example of one site in that data set with, one of the sites we just reviewed a few slides ago. And you can see some similarities already with the biotope. The burrowing and tube-building fauna and the sandy mud are relatively the same at this site. We don't expect to see a great variability in substrate between the most recent sampling in 2018 and 2021 or 2022, just because it's so close. But we could see differences in the biotic components, like Shaina kind of had alluded to, and we're looking forward to continuing the analysis and reporting for the final results, which will be in kind of a dashboard form. And yeah, thank you.
Great, thank you so much, Heather. Jeremy, did you want to add any additional comments, or I think you had mentioned you had some slides you wanted to share.
Yeah, thanks, Meredith. I think I'll start just by kind of summarizing what we heard and making some links between these two studies. Then I have a few short slides I'll show if there's time remaining. So I'll start by just posing the question, like, why study benthos? And Meredith, you didn't completely answer this question. For those of us who are interested in benthic ecology, we don't need any further encouragement. But I think Heather touched on a few things. And one is it is the site of legacy pollution, certainly in the Upper Bay, whether it's heavy metals or also nutrients. And so it is a store of legacy pollutions that could affect the water quality and the fauna. And also it's important that it's the base of the food web. I'll come back to that later. So that's why I think it's important to study changes in the benthos. So I'm still digesting a little bit, but you notice that both studies used a set of tools to sample benthic communities. Both leveraged present-day samples by comparing them with historical samples. So obviously it's useful to go out and continue sampling, but if you can compare them directly with a time series or historical samples, then that greatly increases the value of the samples. And I think you saw examples of that. So starting with the study described by Shaina, core samples were collected by divers at the same locations as historical samples. And Shaina perhaps didn't have time to fully explain the sampling, but it's very sort of laborious and high resolution. These are sediment cores that are collected by divers who go down and take a small replicate sample. And so the quality of the data really relies heavily on the skill with which the divers collected those cores. That's why we have such good data. We also added the beam trawl toes to sample the epibenthos because the cores only take a small section of the infauna, and we want to extend to the epifauna. So I think the main finding, one of the main results that Shaina showed was that the Upper Bay, the Providence River, the community there has become more similar to the Mid Bay or north of Jamestown Station over time, which suggests an increase in successional stage. And Shaina also made the case that nitrogen was the primary driver of the differences in the community. So I just want to reiterate that because that actually I think is one of the most important findings of our study. And I think that was what you showed on slide 28, Shaina. And like we know that the nutrient input has decreased, that's measurable. We also know that water quality has improved in the Upper Bay because the water is clear and people can go swimming and do other things. But I think this is one of the first pieces of evidence that the nutrient reduction has resulted in a direct effect on the biological communities. And that's been very difficult to demonstrate. We know it happened, we know it was very expensive. It's been difficult to actually demonstrate that there's an effect on the ecosystem. I wanted to emphasize that point. By contrast, the benthic communities in the Mid and Lower Bay have changed much less over time, and it suggests less of an effect of nutrient reduction and probably a greater influence of oceanic forcing. The nutrients that actually come into the Narragansett Bay from Rhode Island Sound are probably equally or more important, especially for the Lower Bay when you get down to our Lower Bay station. So a limitation of this study is that our sampling was really, our inferences are limited to the 3 points where you took the samples, because we have to sample those intensively, and we can't generalize the whole bay. We can only say what happened to that location. And I think that the TNC study, by focusing rightly on the upper bay, where the effects of nutrient reduction were expected to be greater, and I think that's the case, but they have a spatial array of stations that don't allow generalizations to be made about the upper bay. So I'll look forward to seeing those results, and I think there'll be some very interesting comparisons made with the faunal composition at the sites you sampled all the way up to the Seekonk River and our Providence River station. And the TNC study also used photography to show what these habitats actually look like, as opposed to the multivariate data analysis that Shaina showed. And as ecologists, we're very much familiar with multivariate data and NDS plots, but I think it's important also to complement that with actually seeing what these habitats look like. And yeah, I think, do I have a few more minutes? Yeah, okay. So I'm going to share my screen now, if I may, and just show you, wait a minute, cue this up. Okay, let me share my screen. Yeah. Here we go. Good. Okay. So I want to take this discussion beyond the benthos and making the point that these benthic organisms in a shallow estuary like Narragansett Bay are really the base of a food chain that extends to shellfish and fish species. So I have a few slides to illustrate this. This one is courtesy of Pat Barrett, who runs the dredge survey for Rhode Island DEM. And he shared these data on quahog abundance, and you can see the different color-coded regions of Narragansett Bay, including Greenwich Bay in blue, Providence River in red, and Narragansett Bay proper in green. And you can see the average abundance of quahogs in those areas over time. And while you notice that in Narragansett Bay proper and also in Greenwich Cove, quahog abundance has been fairly steady over time, you see an increase in Providence River since around 2012, 13. And so we can't really say directly that, oh, the water quality is improved and quahogs are doing better, because it's known that quahogs can actually tolerate fairly high pollution loads. But certainly the quahog abundance in the fishery in the Providence River has improved over time. So I think that's a real positive as far as benthic communities. And this is a case where the fishers, the quahoggers, are actually happy about this, and they see a positive development in the benthic communities in the upper bay. So that's the quahogs. And we could also look at what we see in the GSO fish trawl. So I've added these stars to show where we sample off of Fox Island and also off of Whale Rock, and just look at how those communities of benthic species have changed over time. So if we look at the benthic species we see in the fish trawl, which includes fish and shellfish, I thought I could get away with showing another NDS plot since Shaina showed it so nicely in hers. But if you look at those benthic communities in the fish trawl, you can see there's this strong decadal pattern, starting from 1960s over here in blue, 1970s, 1980s. Then you see this big shift in the community in the mid-1980s, of 90s, 2000s, 2010, and 2020s going up here. And so the main points here, we see a big shift in the benthic communities. It happened in the mid-80s. And if we were to interpret these axes, we might say, well, this is kind of like time. This is maybe climate change going from left to right. But there's also another axis, a second axis. We might think, well, we could attribute that. Is that a reduction in nutrients? I wouldn't want to speculate now, but that's something we'll follow up on to try to explain, in addition to climate change, what is causing the change in these communities. Now that's kind of an abstract plot, so I'll go back to looking at the species that are actually accounting for the main differences between the communities. And these are the species that are really changed the most over time and account for most of the dissimilarity between the early and the late data. And I've color coded them so that the red ones are what we would call cold water species. And you can see declines, particularly winter flounder, ocean trout, sea stars have virtually disappeared from Narragansett Bay. Cunner is much less abundant. And then a few species, both fish and invertebrates, that have actually increased relative to the earlier times. And so, we attribute most of this to climate change and the change in water temperature, the replacement of cold water species with warmer water species. So I think that's really all I want to say. Our analyses of the longer term data will continue, and we'll hope to report back on that on a later date. So I'll stop sharing, and I think, we're right on time here. So thanks for your attention. I'll turn it back to you, Meredith.
Great. Thank you, Jeremy, for kind of summarizing and emphasizing some points. I know I have a few questions, but before I abuse my moderator position, I do want to pull some questions from the Q&A. So again, please feel free to ask your questions using the Q&A option below, or raise your hand and I can just unmute you if you wish to ask directly. One of the questions, though, about the benthic fauna on the North Jamestown site is that the question is, do they still meet the criteria of a late successional stage, and what are considered the drivers for a shift, for the shift in community composition at just the North Jamestown site?
So I kind of already have an answer prepared. I think it's Rich Bell. So hi, Rich. This is a great question. I would say this is more a question of what organisms have been historically found and what organisms are still being found today at north of Jamestown. Historically, north of Jamestown is considered what's called a nested nucleus-based community. So you're looking at nucleolity bivalves and nested polychaetes, and those are the main, base of what's considered a normal or high succession community in the Benthos. And I actually do you mind if I share my screen, Meredith?
Yeah, go for it.
All right, cool. So what I'm going to show is a table. And so this is a table of the top 5 macrofauna species by biomass, but first I'm going to show you abundance. So, and Jay, it's not New Jersey, it's north of Jamestown. So what we have is a clear domination of medium mastus Amosetta when it comes to abundance, but the second most abundant species is Nucleola proxima north of Jamestown. And then I also want to point to our biomass table, and it's quite different from what we see in abundance. And actually, the most dominating species in biomass in north of Jamestown is Nephthys incisa. So what we're seeing is not, of course, medium mastus Amosetta is the most abundant worm and species in the benthos of the bay, if you're not considering nematodes. But barring that, we see Nucleola proxima as the most abundant species and Nephthys incisa as the highest biomass species. So we are still seeing these two species that have been indicators of normal succession in north of Jamestown historically, as the most abundant and highest biomass species in north of Jamestown still. So I would argue that the successional stage hasn't shifted very much, but when it comes to year-to-year differences in north of Jamestown, they are small, but I think they're more variation of larval settlement and less variation with nutrients or temperature. And I hope that answers your question.
I kind of just want to talk a little bit about, or have a question about that successional kind of assembly. And I don't even know if this is an appropriate comparison, but I kept thinking about like when there's a forest fire and it clears out, and there's like a series of steps for it to mature back. And yep. I guess I'm trying to understand more clarity about what successional stages mean, and if we have like a reference for like, or would you even define a benthic community as more mature and defining of, I guess, some of our goals. I don't want to say like healthier or recovery, because I just
Right.
shifting baseline.
Yeah.
It's kind of like, how are we defining things? But I guess I'm trying to understand, what would we consider a mature benthic community? And is that something that you think about in this kind of context?
Sure. So as I mentioned, and I like your comparison, just because that's usually the comparison I try to convey is, when it comes to succession, normal succession after a wildfire. Yeah, I really like that, Meredith. So it's similar to benthic succession where, your baseline, and again, this is something that shifts year to year just because you have different larvae settling, and it also depends on like what species have kind of anchored themselves in the benthos. As I mentioned, longer lived, larger and less abundant species are what you're looking for in a higher successional stage. And so it's not necessarily the species themselves, although certain locations will have indicator species. Like I said, from Jamestown, and actually Boston Harbor is considered a nested nucleus as well. You have baseline species that you normally would see in a normal successional stage community at that site. And obviously those change based on location and climate, but again, it's more the size of the species that you're seeing and the abundance. So if you're seeing these, especially tube dwelling species at the surface of the sediment, for example, four-eyed amphipods, Ampolisca, Heather had mentioned them. And then the other ones are tube dwelling polychaetes. Medium mastus is, like I said, the most abundant species in the benthos, in the bay, worm-wise. But that's mostly because they're second order opportunistic species, so they can colonize anywhere without much hesitation from like hypoxia or what nutrients are in the sediment or heavy metals even. And the image that actually both Heather and I used when it comes to succession is actually from a paper mill. So it's really showing a serious disturbance where like the point where sediment isn't even accessible to be settled because there's just a shroud of carbon that can't be, dug through to settle into the sediment. So we're not looking at that kind of disturbance here. We're looking more at a nutrient disturbance that, has decreased the level of oxygen in the sediment. So again, it's not it's not really even as disturbed as what you would see, from like a paper mill disturbance. But where was I going? So it's yeah. So again, it's like it's a pretty general term for succession, just because it's so varied what you're going to find in the benthos, meter to meter even. But generally, you're looking at high abundance, short-lived, opportunistic species in a low successional area. A lot of them are tube dwelling. And then in a high successional area, you're seeing larger, longer-lived, and deeper dwelling organisms. A lot of bivalves, because some bivalves can live 2,400 years. And then the same goes for our worms. So nested worms can be pretty long and thick. And so you're going to see larger worms burrowing deeper into the sediment at high successional stages, and they can live through the winter. So you're not seeing a constant varying reshuffling of larval settlement. You're seeing the same organisms year to year.
So that's really interesting. Yeah, like it kind of segues into this next question that someone brought forward about, okay, so we have this idea that with succession greater, you're looking at larger, lower abundance, but they're kind of going deeper into the sediment. But the bay has accumulated years and layers of human contamination. We know like heavy metals from Upper Bay, Providence River. So is there kind of a counting for that, or is that like a future study incorporation aspect?
So that question has come up a few times actually, and it's a really interesting question because you have these relics just hidden in the sediment that have been buried, heavy metals especially, and if we are going to have deeper dwelling organisms and when I say deeper dwelling, I mean down to 10 centimeters, because anything lower than that is probably an anoxic zone or getting towards an anoxic zone. So the deepest you're really going to be going down is about 10 centimeters. But if we have organisms burrowing that far down, and we have, a layer of sediment that contains heavy metals, they're going to overturn that and bring it up. And I mean, odds are, since it's not a continuous, depth deposit of heavy metals, we might see, a peak, but then it's going to be flushed out into the bed. So, or it might resettle into the sediment. So it would be really interesting if we could get some sort of, benthic flux measurement, especially in Providence River, to see if, there is this overturning of heavy metals. Yeah, just because I think the best way to be able to measure that would be some sort of like benthic flux, and being able to like target heavy metals somehow.
Great. Thank you so much for that. The next question, is about the TNC study of, I guess, how much change do you see between your study and some of the historical data that you've mentioned? I mean, we know that you're still processing the results, but can you speak to that?
I wish I could speak to it more. I think this is also from Rich, so thanks for your question. We can't say that yet because we're still going through a lot of the imagery. I think, like I mentioned, like a lot of the substrate data, especially from the SPY data set from 2018 and even 2008, is probably going to be pretty similar. However, I think some of the interesting things that I have seen, at least using a higher spatial resolution with like the transects and the drop camera, is getting to see things like lugworm spawning areas that you might not capture in like a smaller resolution, like just one SPY image, which have huge implications for like striped bass, for example, because they love lugworms. And also seeing like soft shell clam beds up in the Seekonk River that are like they die off during the summer, but they do have a resident species or resident population that comes back, which may not be captured again in some of the successional stages that you would get from just like one smaller image or one like core sample necessarily. And then also mantis shrimp that we're seeing up in this like Stillhouse Edgewood area, which, like Shaina had mentioned, represent kind of a higher successional stage. So yes, maybe, but stay tuned. And I'm sorry that I can't share that today.
Well, as your results, as you get them, I'll be sure to try to link them to our website as they come aboard and share and help you share those as you get them. I was actually really curious about your field methods, because they're just so image based. And again, this is not my field of expertise, and I'm just wondering if I'm curious, when you're looking at historical data, kind of how the methods, like the different kind of data that you're working with when everything's so image based right now, and kind of like, are you working with new types of technology, or do you see these kinds of methods being a framework for moving forward to study benthic environments? And that just might be a really naive question, but I find the tools that you're using really interesting.
I think that's where the CMEX standard, the Coastal Marine Ecological Classification Standard, comes into play, because it can be used on different methodology. And so it's kind of taking an imagery method and these other samples and putting into that a more uniform way of comparing them. Obviously, that's not going to be perfect, and there are certain levels that you can get to with that standard that are different based off of those methods. I think one thing that the imagery that I'm excited about is the ability to do some type of machine learning. I don't think we're there yet with these like larger, just like trying to gather data on all the different organisms that are down there. It's already used for like some like coral reef surveying, or looking for specific species. So again, it's really hard, especially with those like muddy environments, for a computer to analyze and be able to tell differences yet. But I think that takes a lot of time and a lot of personnel time to go through those pictures, and with machine learning and imagery, that could really speed things up and make those comparisons more directly, make them more directly comparable. I don't know if that answered your question, but.
No, I mean, well, actually, you elaborated more. I wasn't even considering the use of AI, but I appreciate you even addressing that, because yeah, I just feel like the technology and kind of the ability that researchers would have in the field is going to be able to change so rapidly. I mean, hopefully in a cost-effective way. Otherwise, I guess it won't. But yeah, it wasn't an intuitive approach for me, just thinking about sediments and those kinds of ecosystems, and then all the imaging. I'm like, oh, I guess, it just wasn't the first thing that came to my mind. So that's why I thought it was interesting. Jeremey, I just kind of want to punt something over to you when you're emphasizing the importance of the benthos as the base of the food chain, and what Shaina had mentioned about kind of like the mature successional stages. How, can you infer or use some of those higher level trophic levels of like different fish species to kind of infer different benthic conditions? I guess how much do you, like, if you could elaborate a little bit more on that and what we're seeing within the bay?
Yeah, thanks for the question. And I guess we'll start out from maybe the opposite direction. Recently, we've parameterized the food web model for Narragansett Bay. And well, there have been a couple of iterations. Gary Byron did one. We did one more recently for a project. Where basically it includes all the trophic levels. And I would just say that the trophic compartment that we really know the least about or have the fewest measurements is the benthos. So I think studies like this, and that's why we went to the trouble of estimating biomass, really helps us to constrain the inputs to these food web models. So that's of great importance. You were trying, I guess, well, you were suggesting we kind of do it from the top down, like infer what the benthos is like from looking at their consumers. To some extent, we do that in our. We did that with the striped sea robin by doing a food habit study by looking at what they eat, and so you can make inferences about the benthos from looking in their stomachs. And we do see some of the zonation in Narragansett Bay that we've already discussed and you expect. So yeah, so I guess I would just say a combination of food habit studies and also direct sampling.
There's a question related to the lobster population in the bay, with the idea that it's decreased over the years. But is that correct in thinking, like, what do we know about the lobster population in the bay and its correlation to this kind of work?
Is that one for me?
Not me.
Yeah, I think that could be the subject of another seminar. But the lobster population in the bay has undergone peaks, and the reason why it appeared to be more abundant after 1985, there was a peak in the 80s and early 90s, after which the abundance has fallen off dramatically. So I think if the question was about what's the present status, the abundance of lobsters in the bay, and we can document this in our trawl survey, is very low, an all-time low, and it's been suggested that it's not suitable habitat anymore, and the main constraint is probably the temperature regime. The temperatures in the summer are just above that thermal threshold for lobsters.
Yeah, I actually wanted to ask a little bit about the seasonal aspects of it. I think, Shaina, you had a slide on that, or referencing that earlier in your presentation. With the idea of stratification being harsher in the summer times. And so this is more kind of towards the environmental variables. When you're looking at changes in these kind of communities, from what I understood from that slide, and I could have misinterpreted that, but it seemed like the summer changes were maybe a little bit stronger just based on that stratification if when you're looking at temperature as a secondary component to this, but I guess like how are we really incorporating or viewing the seasonal changes that are either driving these different factors?
And you're specifically talking about environmental variables or how they impact the benthos?
Both.
Okay, so and this was actually something I hadn't and I probably should have reiterated or iterated, but did not. It's important to sample the benthos in the summer because that's peak abundance for the benthos. So when it comes to seasonal changes, and another thing I didn't mention, and I think Jeremy had touched on, the methodology for sampling the benthos is laborious and expensive. So and it's very difficult to do it, especially with cores. You can't send divers down every week to get cores. It's just not, it's not possible. But you can send them down and target, peak abundance in June. And even doing it year to year is difficult. But there was one study in the 80s, I believe, that did a month to month grab of the benthos. And most of what they found was medium mass. So you're not really even seeing the organisms in the winter when it comes to, seasonal changes. And like I mentioned before, longer lived species can survive through the winter, but most of them aren't surviving that, the seasonal changes that you're talking about. So it makes more sense to target the summer for benthic communities. And so it's not there. Of course, it can't really be a continuous timeline of data when it comes to the benthos. You have these points in time that you kind of have to take and compare to. And so seasonality, I think it's really more looking at summer. But I don't know how much. I think that also seasonality affects more the epibenthos, because like I said, a lot of the stuff in the sediment either dies or, hibernates over the winter. So you're not really looking at, although there is also there's a peak of available food right after the winter diatom bloom. And so that's actually what fuels our peak abundance in the summer for the benthos. So you could argue that there is seasonality in phytoplankton, which impacts the benthos. Although I will say that chlorophyll concentration didn't seem to have much of a correlation when we put it into our best analysis. So I can't really speak on that. But yeah, I would say when it comes to seasonality, we're mostly looking at summer peak abundances for the benthos. But there can be impacts from other parts of the food web, like phytoplankton. Does that answer your question?
Yeah, and I'm actually really glad that you brought up the chlorophyll and phytoplankton component, just because there have been conversations over the years about the reduction in nitrogen from wastewater treatments. While we can say, oh, improved water quality, and I had in my notes, I'm like, well, how are we defining water quality in the way that we want it?
Mm-hmm.
Right. And there's some people that feel that if we're reducing nitrogen, we're going to be impacting those phytoplankton blooms that can then and kind of trickle down. Everything's connected. And so your point about that you didn't find chlorophyll, but I don't know if that is tying into the phytoplankton blooms. I might just be making that assumption, but wasn't as much of a determinant here. Is that correct? Did I hear that correctly?
Yeah, that's what I'm saying. It just wasn't correlated with our abundance changes, or community changes, I should say. So community composition didn't seem to be correlated at all with chlorophyll concentration changes, which was interesting to see because you would think there would be a little more of a direct correlation to that. I will also say when it comes to affecting blooms, you're going to get more diatoms if you have less nutrient, if you reduce nutrients. And I could be completely wrong about that. I'm not a phytoplankton ecologist, but I'm pretty sure you get longer lived diatoms if you reduce the nutrients, because what becomes the dominant phytoplankton when you have over production is dinoflagellates, if I'm not mistaken, and again, I could be completely wrong, but that's what I remember from biological oceanography is like you're actually improving the diatom bloom when you reduce nutrients.
Great, nice, thank you. I don't really see any more questions, but I guess, you know, as a last piece, if all of you wouldn't mind just repeating or reemphasizing kind of some of the next steps that you would like to see, not only in like your particular research, but just this benthic ecosystem functioning and like the health of Narragansett Bay, just so we can all leave like with the refresher of like, all right, this is where we are, but here's where like we want to get to within within the realm of research.
I think Heather should go first since her research is like continuing.
Sure. Finishing the project would be no. We're working on getting through that image analysis, so obviously that's top of mind for me. But I think, and I said this before, and it's always been a challenge, but like, and we've brought this up a couple of times during this discussion, but the combination of all of the different methodology and like working together and collaborating with all of those data, you know, there's a wealth of historical data, but there's also a ton of people who are working on, you know, trying to understand this. And I think I get really excited when we have talks like this, and also just like hearing about what other people are working on. I think a lot of time we are, you know, looking down and working on our own work, but collaborating together and like sharing more widely and making things more accessible is something that I would really love to see. And we can always do like that, can always be something that we're working on. So I'll stick with that one.
Dana, any thoughts for you?
Yeah, I completely agree with Heather. I actually, this coastal lecture series was how I realized Heather was doing this work. So it's really cool because I had always had an interest in SPI, you know, camera work, and I, you know, just didn't have the time to incorporate it into my master's thesis. But it's really cool to see that someone, you know, took up the SPI camera and was like, and, you know, is aware of the historical SPI work and is, you know, continuing it. I would say it would be really cool to incorporate all of the historical data and, you know, the continuing, the current data into some sort of database. It would be cool to have some sort of database of benthic organisms in the bay. That would be an interesting goal to see. It would be interesting to see continuous benthic sampling, even if it's just every 5 years. I believe EPA was doing something like this with NCA. I'm not sure if that's continuing. I know it was postponed for 2020. And I know that DEM does dredge sampling, but it would be interesting to see some sort of like long-term standardized benthic sampling in the bay.
Yeah, that makes sense to me. Jeremy, do you want to reiterate some of your previous points or expound?
Sure. Yeah, I'll just make a couple. I mean, there is this time series analysis of the survey data that Connor McMahon's and I are undertaking. So that's a loose end that we intend to wrap up. But I think moving forward for new projects, I think that one thing that these studies emphasize is the spatial differences between upper, mid, and lower bay. And so we can see that that's really distinct. And also that the nutrient reduction has, I think, demonstrably improved the water quality in the upper bay. There's still some residual questions that you mentioned about, you know, the production levels in the mid and lower bay. I think what will help answer that is to actually take forward our food web models. And so this would be a new project, probably done by somebody else. But first of all, to put in the benthic measurements at the bottom to drive it with actual data rather than kind of extrapolating from other estuaries, and also to make that food web model actually spatially determined rather than modeling all of Narragansett Bay together as we've done in the past. So a spatially explicit food web model, I think, would be very useful.
Great, thank you. Well, I think with that we can adjourn unless anyone has any other questions that they want to throw out last minute, but I really do want to thank all of you, our speakers, for taking the time to put together your presentations and sharing your results with us this afternoon. It's not always the easiest work, but definitely appreciate the effort. I think your findings are super interesting, and Heather, can't wait to find out what you come up with. And yeah, we'll be in touch to make sure that I can help you spread the word on those results. I also want to thank everyone who joined us to participate in today's discussion and share conversations. And I hope everyone, took away something a little bit. And Shane, I really liked when you're like, oh, I didn't know Heather was doing this. And I was just like, perfect. That's exactly like one of the main goals of this series. So that's super great to hear. But yeah, hopefully there can be more connections made, further conversations generated, and we can keep supporting this kind of research. But again, thank you to you speakers for joining us and to you participants for also engaging. And I hope everyone enjoys the rest of their week.